Last time we went over the very early development, just the initial, so there was, you form a blastula, which is a sphere, and then there's an in-pocketing, that's gastrulation, and then that leads to the formation of the neural plate, which is the top layer of the in-pocketing, and then the neural plate rolls up into a tube, and that's the neural tube, which is the sort of the basic tubular structure of the nervous system. And so what we want to talk about today is somewhat later development. So not too late, but just where are the main front to back segments of the nervous system, because the nervous system, like the body, is segmented, and this is similar to the segments in a worm. So the point of doing this is that you have some kind of sort of architecture on which to mount the various neural structures, like where are they relative to each other. And when you have this very simple schema, it gives you some, when you're reading neuroanatomy of the adult human brain, it's just an incredible mess. Everything's all twisted up, and it's sometimes hard to keep track of where a particular piece of the nervous system belongs in the overall scheme. So that's really the goal today, to see how we can go from the neural tube to the way the brain looks as an adult. So I always start off with a basic schema for the neural tube, and I almost always ask you to reproduce this on a test, because it's such a basic thing. It's worth memorizing this. And so if we start off at the back of the neural tube. So we've got the spinal cord, and the spinal cord is a pretty big part of the nervous system. The spinal cord itself isn't very big, but it's quite long. And so there's a lot of circuitry in the spinal cord. Here's the ventricle in the middle of the spinal cord. And remember, that's the same ventricle from the original inside of the tube. And so that ventricle is going forward, and here's the sort of anterior extension of that tube. And so what happens in later development, after the tube has closed, so after the tube has rolled up and closed, what happens is it opens up again. So here's the ventricle, and then what happens is there's an opening that develops like this in the ventricle. And so what happens? So what happens is so he starts off with a neural plate, it closes, seals shut, and then after it seals shut, it opens up again. And that thing is called the fourth ventricle. Now that sort of implies that there's more than one ventricle, but in fact, there's only one ventricle. All the ventricles are the same ventricle, and they're just the inside of the neural tube. And so this is the fourth ventricle, and as a result of additional studies, we know that the segmentation of the neural tube is a little bit more finely divided than I'm going to draw right now. But what I'm drawing right now is are kind of generic front to back regional differences in the neural tube that are you'll commonly see. And so since we have a cylindrical coordinate system, what are the coordinates of it? They're slightly Cartesian. So it's sort of, so one coordinate system is one direction is rostrocaudal. And what does that mean? So rostrocaudal means rostrum is the nose, and caud means tail. So this is sort of nose to tail. That's a good coordinate for a cylindrical coordinate system, because there's nose going down to tail. But the problem is, part of the problem with that is the fact that if you look at a quadruped, the brain kind of looks like this. And so and then here's the nose. So there's the nose, and here's the tail. So in that case, sort of rostrocaudal makes sense. But if you look at a primate, especially humans, bipedal primate, what's happened is the spinal cord is like that. That's a different. So the brain is about in the same place, and the eyes are, here's the eyes, looking forward. So there's the eyes looking forward. But the spinal cord has sort of been bent by 90 degrees. And so sometimes in human anatomy they say, anterior posterior. And anterior posterior is kind of like rostrocaudal as far as the brain goes, but it's a little funky, because you also have ventral, which means stomach, and dorsal, which means back. And so if you look at a quadruped, here's dorsal up here and ventral, and you can see that the dorsal ventral direction is kind of perpendicular to the rostrocaudal direction, but in a human, the anterior posterior is kind of the same as dorsal ventral. So the terms get slightly confusing. So then we have dorsal ventral, but in a human brain, a lot of times instead of dorsal ventral, which makes sense in a quadruped, we say superior. We often say superior inferior, because that's actually that was the quadruped. We say, superior, inferior, because superior, inferior sort of looks like rostrocaudal because of the 90-degree bend. So but if you look at any sort of animal studies, they'll often use dorsal ventral, back, stomach. And then there's another one which is mediolateral. And that one's a little confusing, because that one's kind of like a mirror image. So mediolateral means left-right, but it's kind of like, medial is on the middle, and then, lateral is going out either way. So it's a little different than the dorsal ventral in the rostrocaudal case. So there's, rostrocaudal, dorsal ventral, mediolateral, superior, inferior, anterior, posterior. Mediolateral is the same for humans, because nothing, that 90-degree bend just occurred sort of in the sagittal plane. So back to the nervous, back to the neural tube. So what's here, at the fourth ventricle and behind? So if we divide things up, the first division is often just conventionally at the back of where the fourth ventricle just starts to open up. And that's, generally called, the spinal cord is behind that, and the brainstem is ahead of that. And then we divide up this. There's a big boundary here, which we'll talk about, called the pons midbrain junction. But there's a conventional division into the pons and the medulla. And so, like I said, there's actually more segments in here if you actually go back and look at the way that the segmentation specification of the brain works. But these are in the literature, so the pons and the medulla. And there's one last thing here behind the pons midbrain junction, which is the cerebellum. So here's the cerebellum. So there's the top of the fourth ventricle and the cerebellum. So this is cereb. Okay, so these pretty much have, you've got, we'll talk in more detail about what are the main pieces of hardware in the pons and medulla. But what's with this pons midbrain junction? So this thing is the pons midbrain junction. That's an important rostrocaudal dividing line in the brain. And this involves what I call the rule of Sereno. So unfortunately, this isn't sort of taught in neuroanatomy classes. It really should be. So what happens at the pons midbrain junction is that the left-right specification of the brain is flipped. And so let's draw what's in the midbrain. So if we go up to the midbrain, there's two bumps in it. And since there's a left and a right copy, in the human it's called, the corpora quadrigemina. So it's like the four-part body. And so what are those parts? There's the superior colliculus, and then behind it the inferior colliculus. So superior colliculus, inferior colliculus. So that's the midbrain. So here's the top of the midbrain. This guy's the midbrain. So what's with the pons midbrain junction? Well, if you look, I didn't draw the ventricle in. So the ventricle closes again. So there's a ventricle in the midbrain. It's the same ventricle right here that we opened up and we could see down to the bottom of the underside of this. You could see down to this part down here. So now it's closed back up again. So we have we basically have a neural tube again. But what's the significance of the pons midbrain junction? Well, if we look at the eye, so the eye is over here. It's past the pons midbrain junction. So how does the eye connect up to the brain? Well, the eye goes and crosses over, and it connects over to the right eye connects to the left superior colliculus, and vice versa. And that contrasts with the ear. So the ear is down here. Here's the earlobe. So the ear just connects up to the same side. And so the right ear connects up to the right brain, left ear connects up to the left brain. So what's the sort of origin of that? So as far as we can tell, the origin of that is that it's probably related to the fact that the eye, that the lens, or the cornea really is the main lens of the eye, actually does a left-right flip of the image of what's out there. That's one plausible explanation for why this occurred. But basically what happened, and it must have occurred very, very early, at the time when the eye was very crude, could just basically tell sort of, something over here, something over here. And it gets a little more complicated in primates and a lot of mammals that have forward vision. We'll explain some of the details, but this is the primitive condition. The right eye connects entirely to the left side of the brain. So that probably had to do with this developmental respecification of the kind of what's left and what's right, and so that leads to the rule of Sereno. And so what is the rule of Sereno? So the rule of Sereno, so we'll put it over here, rule of Sereno. So basically this explains the laterality of 95% of the connections in the brain. And so if here's the midline, and here's the pons midbrain junction. So this is pons midbrain. The rule is if you're in the body, and here's the body out here, you go to the opposite side. If you're past that junction, if you're in the body, you go to the same side. But it turns out, so that's what I already just drew here, but it turns out almost all of the connections in the brain respect this left-right switch. So if you're on the right side of the brain and you connect up to another structure that's on the same side of the brain, you generally will be uncrossed, or we would call it ipsilateral, same side. On the other hand, if you're in a part of the body and you connect to the opposite, you go past this junction, not connect to the opposite, go past this junction, what will happen is you'll switch over and connect to the opposite side of the brain. And similarly, if you're past that junction, generally you're going to connect to cells that are on the same side of the brain. And the same thing would occur for a projection that went up. So if something comes up from the spinal cord and it's going to go up to the thalamus, it's going to cross. So exactly where it crosses is not as important as which side of the brain the source and the target are on. So sometimes the cross doesn't occur right at the pons midbrain junction, but what will happen is, axons will come down. They'll maybe stay on this side for a while, but not connect up to anybody, and then cross over and start connecting. So that explains 95% of the variance, or 97% of the variance in which side of the brain connects to which side. Now, you might immediately think, what about the corpus callosum? Corpus callosum connects the left and the right. And so people have spent a lot of time looking at the corpus callosum. The corpus callosum is nothing. If you look at the connections of the cerebral cortex, 98% of the connections, or 99% of the connections in the right cerebral cortex are to other parts of the right cerebral cortex. And you got a little corpus callosum, tiny little corpus callosum. It's 1% of the total connections in the white matter. Tiny little thing. So there's an example. There are some things that break the rule of Sereno, like the corpus callosum. But 98% of the other stuff doesn't break the rule of Sereno. So there's overwhelmingly within hemisphere connections. So corpus callosum is very small compared to all the intra-hemisphere connections. Okay, so let's continue up. So the next segment is called the thalamus. And so there's the ventricle going through it. And the thalamus is, so sometimes a single word like the thalamus is used to refer to different levels of the hierarchy. So sometimes when you say thalamus, you think of the lateral geniculate, which is actually just a subpart of the greater thalamus. It's called the dorsal thalamus. So sometimes people say, if you just want to refer to the greater thalamus, you might say diencephalon. Diencephalon. That refers to the entire thalamus, including the hypothalamus and other parts. And so there's the thalamus. If you go up to the front of the thalamus, there's another opening in the neural tube that opens once again. What happens is the neural tube completely closes, and then it opens after it's completely closed. So this thing right here. So how does this work? Well, actually, how does the cerebellum work? You completely close the neural tube, you open it up, and then the cerebellum grows over the opening. So what happens with this opening? So this guy is called the third ventricle, and that's not a good name for it, because after all, there's only one ventricle. It's the same one. So that's the third ventricle, which is just continuous all the way down to the spinal cord. But what happens here is that the a little sheet of non-neural tissue called a tela forms across this thing, and then what happens is that little thin sheet then sort of protrudes into the ventricle, and so there's another rostrocaudal boundary here. So it protrudes into the ventricle, and what it does is it forms a sort of a Y-shaped structure, and this little thing sort of protrudes into the ventricle, which is more or less sealed off and not very big, and it kind of expands it out like a balloon from the inside, and that's why it's called an evaginated forebrain, because it actually sort of forms by this sort of in-pocketing. And so then this one's called like I can't remember which is which, but there's the first ventricle and the second ventricle. First, well, I can't remember which side is which, but we'll call this first and second. And of course, and then here's the third ventricle. It's the same old ventricle, except now we have a Y-shaped structure, sort of a left-right duplicated structure. So that's sort of the basic sort of scheme of the brain, and this part up here is called the forebrain. Or sometimes it's called the telencephalon. Telenceph. You might have seen that. So that's the forebrain. So now all animals don't do it this way. At this point, I usually spend about 5 minutes talking about fish. So bony fish. So let's look at just bony fish. Like a salmon is a bony fish, as opposed to like a shark. So almost all animals do it this way, like a frog. This is pretty much what a frog brain looks like. The cerebellum just looks like a little patch going across. So if you look at a frog brain, it looks pretty much just like I drew here. Now that doesn't look like a brain to you, I'm sure. It's, if you've looked at a human brain, it doesn't look anything like that. But a frog brain does. But a fish brain doesn't. So what a fish brain, interestingly, decided to do is this thing happens here, where this third ventricle so they've got a fourth ventricle, and the third ventricle forms, but instead of turning inward, sort of evaginated forebrain, and forming it sort of an inward pocket, it turns outward instead. And so what happens is, you start off with something like this, with a little thing across the top, just like this, but instead of then sort of going in, it goes out. And so what happens is you end up with this weird shaped brain like that, something like that. And then what used to be this little thin thing is kind of spread out over the top. So basically the ventricle, so if this is here's the ventricular surface. The ventricular surface ends up outside the brain like that. And it's called, this is called an everted forebrain, which appears in things like a salmon or a bony fish. So a lot of times when you think of a fish, you think, well, I just eat fish. They're just fish. They've got all the same parts of the brain that we do. So you hear people say ridiculous things like, can fish feel pain? Of course fish can feel pain. It's actually really bad if you can't feel pain, because then you sort of damage yourself. So yes, they've got all the same things. But what's it like to be a fish? So most people have sort of not much intuition about what it's like to be a fish. So what is the biggest part of the brain in a fish? So the biggest part of the brain in the fish is where the taste input comes in. There's actually a big bulbous part of the brain, bigger than its forebrain, down here back in the pons and medulla. There's this giant layered structure that takes taste input. So fish can sort of taste. They can taste with their mouth, but they can taste with their face, and catfish can taste with their barbels, and they can taste with their tail. So they can just touch something with their tail, and they have taste buds on it. If you look in the fish brain, you find maps of taste buds. So like visual maps. So you have like tastotopic maps. And if you look at something like a goldfish, You've probably, if you've ever had a goldfish, you've seen a goldfish and they'll suck up some junk from the bottom of the aquarium and then spit it out. And you think they're just like, oh, I didn't like the taste of that. What they're doing is much more complicated than that. There's something inside the throat of a goldfish called the palatal organ. And what that is, so these are the gills right alongside here, like this. So those are the gill arches, those same gill arches we drew last time. Here's the rest of the fish. There's a fin. You got a fin up there. So what is the fish doing? It's doing this unbelievably high-tech, spatial taste-based sorting operation. And so what it does is it sucks in a bunch of crap, and there's a bunch of some good stuff in there. There's like a little potato chip that you dropped into the aquarium, and then there's like a little bit of fish food, and then there's some other stuff that it doesn't want, like a little rock and a little piece of dirt. I don't want that. And then over here there's a little tiny piece of algae. I want that. And so what it does is this entire surface is completely densely coated with taste buds. And also on the gill arches, there's taste buds there. But then in addition, there's little muscles. There's little muscles that are controlling each one of these things separately. And so what the fish does is with those muscles, it hangs on to the little stuff that it wants. So, but in a spatially complicated way. So it'll hang on to 10 things. I just want these things. And so hang on to them with the little muscles. And then when it goes, it's just blowing out all the other stuff that it doesn't want. And then it eats the stuff that it wants. So it's this extremely high-tech, spatial, taste-atopic, muscular sorting system, muscular sorting. That's actually what fish are doing. So what's the second biggest part of the brain in a fish? It's the hypothalamus. And the hypothalamus is actually bigger. The hypothalamus is bigger than the fish forebrain. So the equivalent of our cortex, the hypothalamus, like drug, sex, and rock and roll part, is bigger. And so what's it like to be a fish? I always say this. You're like this gourmand, a very emotional gourmand. You fight with other fish, you eat stuff, you really enjoy it, you get excited, you get angry. That's what it's like to be a fish. Giant hypothalamus, giant taste. So it's probably not what you thought what fish were doing. Okay, so let's erase the rule of Sereno, but I'll leave this thing up here. And so what we're going to do now is basically look at what the initial structure, so this is early over here, and later over here. So now we'll sort of try to make drawings of the parts of the brain as they more or less look as an adult. So if you look very early in the spinal cord, so we'll start off with the spinal cord. So if you look early in the spinal cord, what do you see? You see something that kind of looks, more or less like the neural tube. So you have something, a small thing, I'm drawing it a little bit bigger than it is, like this. And you got your radioglia there. There's all the radioglia, but it turns out there's a big functional division in the neural tube, which goes all the way up to the thalamus, and you could argue even into the forebrain. And what this is the division between sensory and motor. So this part is called the alar plate, the top quarter, and this part is called the basal plate. The plate meaning sort of like a plate of cells, kind of like the cortical plate or the neural plate. So it's the alar plate and the basal plate, and basically this is sensory, and this guy is motor. And that, like I said, applies going all the way up to the hypothalamus. We'll see the same thing in the hypothalamus. So what does that look like sort of in the adult? Well, it's really pretty different in the adult. So you end up with a big spinal cord like this. So, what's in that spinal cord? Well, you've got the ventricle is now just shrunken down to a little thing like that. And then you start to have white matter in the spinal cord. So what happens is you've got something like this. So that's kind of like the gray matter of the spinal cord, and then you've got sort of the ventral horn. So that's the dorsal horn and the ventral horn. So here's the left-right subdivision. I slightly tilted. And so this is the ventral horn. That's where the motor neurons are. So here's a motor neuron there, and that will send an axon out of the brain. It actually just penetrates out of the spinal cord, and it will go and synapse on a muscle. So there's a muscle. So that's a motor neuron. The sensory neurons are those things that came from the dorsal root ganglion. So there's those funny cells. They've got a receptor that sort of goes out into the skin right there. That's one end of it doesn't really have any dendrites. It just has it's just a big axon. It's this axon. So here's the dorsal root ganglion, and then that goes in, and then eventually sort of synapses. Sometimes it goes even in some cases in synapses on motor neurons. So this part here, the dorsal horn, is kind of the sensory part of the spinal cord. So there's a whole bunch of neurons up here that have sensations of touch and length of muscles and force in muscles, a whole bunch of different temperature, pain, all that stuff is up there, including in the goldfish. So that's the adult, what the adult spinal cord looks like. Now, if you look at the medulla, what happens is now the original tube has opened up. So here's that original tube. So it's open. It started to open up like that, and then there's this little non-neural thing that covers over the top, and eventually forms the choroid plexus, which is the cells that generate the fluids that bathe the brain. So that's what. So it's basically just looks like this, except you just opened up the top and spread it apart. And when this all grows up, you end up with something like this that looks sort of more or less. It's got sort of 4 bumps in it like that. But that same division, between like sensory and motor, which is this division here between sort of sensory alar plate, dorsal. So this is dorsal, meaning it goes on the back. That's back, and this is ventrum is the stomach, so there's ventral. So that same division is now sort of like off like this. So this basically is the, so here you have like taste, touch, and you've got some, there's some vestibular that's related to the ear, but those are sensory things. And then if you look down here, you have things that originally came from somites, like the tongue, that's somites. But then you also have things in the throat that came from, gill arches. So remember there was that distinction between those two segmented parts. The somites, which was the segmentation of the spinal cord going all the way up to the eye muscles. And then you have this separate set of gill arches, another segmentation system controlled by a different set of genes that forms things in the throat. So these are both motor. So this is motor here, and sensory up at the top. So that distinction, and then is another motor over here. So this is just half motor. Remember, it's all half. So it's all related to stuff on the same side of the brain. So that's we haven't talked about any of the structures, but that's kind of the the overall shape of things. So what happens to the pons? Well, the pons pretty much looks about the same as the medulla, except it's got the cerebellum over the top. And if you look at a frog, it pretty much looks like this. There's just this little ridge going across the brain right here. It's got all the same cells in it, Purkinje cells, climbing fibers. It looks like the same kind of cells you have in the human cerebellum, but it's just like this little flap. It's not folded at all. So what does that look like? Now I've got to actually scale this whole thing way down, because the parts related to the cerebellum have gotten so big that you can barely see the original neural tube. That's one of the reasons why it's kind of hard to recognize what it actually looks like. But if we start off, we basically have something that sort of looks like this, like we had before, and then there's the cerebellar plate sort of grows. These cells that are sort of sitting right about here, they sort of grow over that little non-neural tissue and form the cerebellum. But what does it look like when you're an adult? Well, it doesn't look anything like this. So if I scale this magnification way down, way in the middle, you've got a little thing that still looks like that. It still has that characteristic shape. You can see if you look at a human brainstem. But then what you've got attached to the bottom of it, you got this absolutely gigantic thing, which it sort of looks like sort of a cut through beef or something, but it's called the pontine nuclei. It's actually divided left-right, but this thing is the pontine nuclei, and it's an unfortunate name. So they called it that because it's kind of in the pons, but it actually acts like the midbrain. So it acts like it's reversed. So the right side of the pontine nuclei have to do with the left side of the body or the left side of the visual field, and vice versa. So it acts kind of like the midbrain, not like the pons. But it completely, you can't even really see this part right here, because there's this giant thing sticking off the bottom. And then the cerebellum in humans is gigantic. It's expanded even more than the cortex has relative to a monkey. And so if you look at the cerebellum, it just billows out into this gigantic thing. And it's got 20 zillion little folds in it. There's almost as much cortical surface area of the cerebellar cortex, as there is of the cerebral cortex, about 80%. And if you look at it, what does it look like? It's got like a million little, they're called folia. They're all these left-right folds. So this is the cerebellum. And if you stretch the whole thing out, it's maybe like 10 centimeters wide, but it's like over half a meter long. So it's like it's about 3 feet long. It's all packed in that little back, so you could stretch it out into this long little strip, because it's all folded in only one direction. And what happens is it extends, the cerebellum extends all the way down, completely covers the fourth ventricle and part of the spinal cord, so you can't even see the fourth ventricle, the opening in the fourth ventricle. So that's what and the cerebellum, it's behind the pons midbrain junction, so the right cerebellum relates to the right side of the body or the right side of the visual field. Left cerebellum relates to the left side of the body. So unlike the pontine nuclei act like they're in the midbrain. Okay, so now let's go up to the midbrain, and so the midbrain, the ventricle closes again. And so when the ventricle closes again, what happens is you end up with something looks like this. So if you go to the superior colliculus, there's superior and inferior, so we'll just draw the superior colliculus. So there's a ventricle, now it's the same ventricle, now it's closed, but it still respects that same division. So this is basically sensory stuff up there, This is sensory and motor stuff down here. So what are some of the motor things? You have things like the red nucleus. Like the red nucleus is the original motor cortex, and you have the substantia nigra down there. So this is the red nucleus that controls the body, and it's in the midbrain, so if it's going to connect to the spinal cord, it has to cross. Here's the substantia nigra. So that's the black substance. It's slightly black. It's got a lot of melanin in it. The red nucleus is slightly pinky, so that's why it's at the red nucleus. And there's a lot of other structures down in here for controlling body movements. And there's eye movement control is in here too. So that's the midbrain. If we go up to the thalamus, again, that just started off as just a little, neural tube like this, basal and alar plate. So what about, going up to the thalamus? So if you go up to the thalamus, we typically divide that into 4 things, top to bottom. But there's kind of like 2 more sensory ones, 2 more motor ones. So if you look at the thalamus, you see something like, it's got like 1, 2, 3, 4 little bumps. 1, 2, 3, 4, like that. And so there's those 4 segments. And what are those called? Those are called the epithalamus and the dorsal thalamus. That's the one that you're probably more familiar with, and the ventral and the hypo. So this is thalamus. Thalamus, this is midbrain. So what does that look like when the animal's adult? Well, it really massively expands out. So the hypothalamus is pretty big. So here's the two halves of it. The ventral thalamus turns into this little thin sheet. It's a little thicker at the middle, like that. And then there's an absolutely gigantic dorsal thalamus. And then here's the little epithalamus. So that's, if you look at the adult thalamus, that's kind of more or less what you see. So here's epi, dorsal, ventral, and hypo. So if you think about what's connected up, this is connected up to the pituitary, and that's kind of like a motor structure. It releases hormones into the blood, and those cause things like you can get you all excited or fight or flight kind of things. The epithalamus, it has a little eye. There's actually a little eye attached to it. It's light sensitive, so it actually receives. It's called the pineal. So the pineal is up here. Not quite. Well, go in a little bit. Pineal. Yeah, so the pineal gland, it's a kind of a sensory structure, basically. So the dorsal thalamus is the one you think of, like the lateral geniculate or the ventral basal, that have all the sensory relays. The ventral thalamus gets a little more motory. So the subthalamic nucleus, that's part of the basal ganglia circuit, that's in the ventral thalamus, related to movement. The hypothalamus really is related to sort of like stimulating the body. So that same kind of sensory, I erased it, but it used to be up here, sensory motor thing, is present in the thalamus. So finally, the forebrain. And the forebrain is the one that expands the most. And so when we look at the forebrain is the one that looks least like what it does in the neural tube. Now, if you look at the forebrain of a frog, it looks just like what I drew over there. But if you look at the forebrain of a human, it doesn't look anything like that. And so how does it get that way? So if we start off, so if we just cut through like the forebrain of a frog like that, what do you see? You see, 2 tubes. So you actually do see two tubes like this. And what are the main parts of those tubes? The main parts of the tubes, so here's the midline. They're mirror images of each other. And so you have the inner, sort of the medial bottom quarter of the tube is called the septum. And then the lateral bottom quarter is called the basal forebrain. And then the top is called the pallium. And some of the other subparts of the top, there's the same. This is a mirror image, but I'm just drawing it twice so I can annotate it on this side. So this is the amygdala, and then this part here is the hippocampus, and then the neocortex comes from this part. And there's, the septum is over here again. So that doesn't look like, certainly doesn't look like anything that looks like a modern brain. So how does it get that way? So it gets that way by basically, so if you look at it from the side, so this is a kind of a side view here. So there's where it Y's off. And here's the ventricle, and the ventricle sort of like is Ying off into that guy. So what happens is you have the formation of the temporal lobe. And so if you look at this and you say, if that's the hippocampus isn't there. You know that the hippocampus is kind of down underneath. So how did it get where it goes? And the reason that it gets where it goes, so here's the hippocampus originally up here. So it's in this dorsal medial position. And what happens is this giant sort of migration of cells like this is what causes the formation of the temporal lobe. So the cells all sort of like this mass migration, they sort of like migrate caudally first, and then they go down, and then they come out laterally, and they rotate at least 180 degrees around. So that's the formation of the temporal lobe. Formation of the temporal lobe. And what does that look like from the top? So if you just draw the same thing from the top, so here's the ventricle, it splits, there's the third ventricle, and then it comes off and goes up like that. So like frog stage. And so what happens is the hippocampus, which is kind of situated here, kind of all rotates around like this. So it kind of, try to draw that in perspective, gets smaller. Yeah, so basically that's what happens. That whole thing sort of, it happens, same thing happens on the other side. So, it goes around like that. Now if you look at a frog, if you look at something like a rat, you can see that this is only partially happened. So let's look at a rat. So if you look at a rat brain, you've got the olfactory bulb. They got a pretty big olfactory bulb, and then there's the cerebellum over here, and then here's the opening of the fourth ventricle. The cerebellum is covering it. There's the pontine nuclei, and there's the brainstem. So that's kind of like a rat brain. If you look at the position of the hippocampus in the rat brain, it's pretty much vertical. So that's H. So it only rotated about 90 degrees. So the hippocampus is a vertical structure in a rat brain. It's a little confusing if you look at rat brain sections, because they'll sort of cut sections like this, and then suddenly you go through the hippocampus, it's vertical. But if you look at a primate brain, that's a rat. But if you look at a primate brain, it's you get something that kind of looks like, like this. So here's the here's the cerebellum, and way buried under there, completely invisible, is the fourth ventricle. So there's the brainstem coming out with this huge cerebellum tucked in there. But the rotation has been more like 180 degrees, and so whereas the hippocampus, it's kind of sort of ends up something like that. So that's H. And so it basically runs into the amygdala, so there's A. But the amygdala came from the other side. So this thing sort of like goes all the way around and actually almost bumps into the amygdala, which it was originally pretty far away from. And so that leads to, all these complicated pathways like the stria terminalis and the fornix and the stria medullaris, and there's a whole bunch of pathways. And so why are these, if you look at a neuroanatomy textbook, it's just trying hard to remember them all. Why are they like that? Well, if you look at the original plan was the hippocampus projects to the septum. And so what does it do? Well, not hard. It just goes, the septum was right next door. It just went to it. To do the same thing here, you've got to start in the hippocampus, and you've got to go all the way around the fornix, and then branch off and get into the septum. It would have been easier just to go straight through. And there are a few pathways that have figured out how to go straight through. Like the amygdala also goes through this long spiral path, but there's another pathway coming out of the amygdala called the ventral amygdalopetal pathway. It's the ventral pathway that flees from the amygdala, and that one just figured out, I'll just take a straight shot through because I need to hook up to the thalamus. So that's the mess that we end up with. And so what does it look like when you actually look at the adult brain? So if I cut a section through the, where's the septum in a human brain? So if you look at, the adult brain, it's got the cortex, this giant cortex sort of billowing out over the whole thing. The cortex goes like from top to bottom. And then you can actually see the first and second ventricle. So here's that ventricle right there. And if you look at that, let me just draw that. So this is the septum right here. So it should be basically on the medial, that's medial, bottom part of the ventricle, and that's where it is. So here's where the septum is right here. It's very small, didn't get anywhere near as big as the cortex did. And this is all the white matter here, but inside the white matter you have all the basal ganglia. So the basal ganglia would be sitting over here like the caudate and putamen. So those things, are corresponding to this bottom, the lateral ventral part of the neural tube. But the top, the pallium, comes all the way around the bottom. So you end up with infratemporal cortex way below the striatum. So this is the septum in the adult, and this is the striatum. But really, this is where it came from. And this is all pallium all the way down to here and up here. So looking at this, and then paste in this kind of all these circular pathways, and your brain explodes. But if you keep this thing in mind, this will kind of give you a kind of a basic coordinate system to sort of mount your connections on. So you can sort of figure out, if we're in the hypothalamus and we've got some, projection to another structure in the forebrain, it's almost always going to be on the same side. And we can sort of remember the hypothalamus is part of the thalamus past the pons midbrain junction. Or say like the red nucleus, it's in the midbrain. So the red nucleus has to go to the spinal cord, it has to cross. If the cerebellum wants to go to the red nucleus, it has to cross because it's crossing the pons midbrain junction. Okay, so David, I'll press that button and we'll see if we can get a few pictures up. So overlay only. Let's see. Okay. So I sometimes put up this this picture about evolution. Humans at a campfire, it's cold, I'm starving, maybe I should ask for some scraps. What's the worst that could happen? It's like a savvy wolf thinking about it. And you got like 10,000 years later, didn't work out. Okay, so let me go and just look at, this is one last picture of the eye. So here's this was a particularly good one. So this is the retina here. This was from last time. So that's the retina. And that part's the pigment epithelium. You can see that here. That's pigment epithelium. And then here's the little vesicle of the lens. So that's the lens. And then you can see a little bit of the skin. That's the little skin closing. And so the eyelid hasn't formed yet, but the eyelid will sort of like where the cells die right there in order to actually sort of open your eyes. So, okay, so that was one I meant to do last time, but I forgot to do. So let's erase that. And okay, and this on the right side here, this is a picture of bithorax. So this is the way that the fly is supposed to be. It's got like one big wing and then one small wing. Like dragonflies have two big wings, but flies are more advanced. They have this little inertial guidance system, the haltere. But here you can see bithorax. So you can see, it looks like it has two thoraxes, and so that was a gene, a mutation in one of these segmentation genes. And here was an example of the antennapedia. So here you can see this leg coming out of, the head of the fly. What's supposed to come out there is the antenna here. So here's the antenna, like that little thing is the antenna. But instead you got a leg, not a good look. So that was an example of these very early genes that basically disturb the basic segmentation, the basic identity of what the segments in the body are. So if we look over on this side, what this is they. Let me escape that guy and pull him over here and blow him up. There we go. So this is the formation of the ear. So this guy is, so here's that in-pocketing that's occurring. You can see that's a typical thing where all these guys are scrunching their hands together and their feet, they're letting their feet sort of spread out to cause an in-pocketing, and then you eventually get a little vesicle. That's the otocyst. It's called the otic placode, and that's what's going to turn into the inner ear. So the different parts of the inner ear. So that's different than the eye. So in the eye case, what happened was you basically had a little bulge out of the brain itself, and then the stalk dies off, and then you hook up to that retina. Whereas in the ear case, what happens is you've got an actual placode that forms, and then from the body. Now, this is a picture of what the actual neural tube looks like in an early human, and so here's this is the ventricle right here. So you can see it's kind of it gets a little bit of a creasing thing going on. So this thing here in blue is basically the inside of the ventricle. So what you're looking at there is the thing I drew like this. So that blue part is this inside of the ventricle that you can see right there. And then it closes up again, so the ventricle is now closed, and then it goes into the forebrain. So there's the split in the forebrain. So here it's open, and then here's the very back cut edge of the of the fourth ventricle. So here's the very back where I drew this a little wrong. Let me correct that. Yeah, so let's get rid of these guys. Okay. So basically here's the ventricle coming up like that. And then now it's open, and then it's closed again once you get up to here. So yeah, it's a little more folded up than I drew. That's a little bit later in development. But here's another picture. This is a good picture of it from the back, so you can really get a feel for what it looks like in 3D. So here's the ventricle closed, and then here's that little bottom of that little triangular thing that I showed where you can now see inside the ventricle. And it's got a little folding going on. This thing right here, the rhombic lip, that's where the cerebellum comes from. So the cerebellum sort of grows over the ventricle and eventually completely covers the ventricle. Like in a human, it covers the ventricle down to here. But then right about here is right where the ventricle closes up again, like that. And so here's that cerebellum that, look at your pictures. That is the rhombic lip where it comes from, but there's an extra fold. So the cerebellum has grown around like that. So it comes out of the rhombic lip. That's where the cerebellum comes out of. But it's already started to fold up a little bit. It sort of, grows in that direction and this direction. And then when we get up to here, this is where the superior colliculus is. So the inferior colliculus is going to form sort of like about under there, and this guy is the back of the superior colliculus. So that's it doesn't look quite like the very simple tube I was showing you. But if we go back to something like a frog, here's a picture of a frog, and so here you can see that it does look like I drew. So you can see, here's that opening in the fourth ventricle. It's closed right here. Here's the superior colliculus, and then here's that the pineal body sort of covering the fourth ventricle, the third ventricle, and then here's where it splits into the two Ys. So that's pretty much like that diagram I have on the left side of the board. And that's an adult frog. So, okay, so that's it for that. We'll stop and take some questions if anybody's still there. Probably not.